The topic of chapter 13 is blood, the heart, and circulation. Blood has a number of different functions. The first is transportation. As you know, the cells within your body generate CO2 as a waste product of aerobic respiration, and your cells need oxygen. So oxygen is transported in the blood as well as CO2. Your cells also need nutrients. So the nutrients, for example, things like amino acids and fatty acids and glucose are transported throughout the blood, and those are received by your cells. And the cells of your body also produce wastes, for example, nitrogenous waste such as urea. So those are transported through the blood and are excreted in the urine. Blood is also important for regulation of your body. So, for example, hormones are released by endocrine glands. They travel throughout the blood, and they have an effect on cells that have specific receptors for those hormones. Temperature is also another important function of blood, so temperature regulation. For example, the inner core of your body is hotter. It has a temperature of about 37 degrees centigrade, whereas the skin is quite a bit cooler. And so in order to bring heat, in order to dissipate heat to the environment, your body is designed to bring blood from the core to the surface. So it brings it to the skin, which is highly vascularized. And this is particularly evident if you're exercising. You'll notice that the skin becomes flushed, and that's because of vasodilation, which is an increase in diameter of blood vessels within the skin. This raises the temperature of the skin. There's therefore a bigger difference in temperature between the skin and the environment, and this allows for greater dissipation of temperature, of heat from your body to the environment. And exactly the opposite process occurs when you're in a cold environment and you're trying to conserve your core body temperature. Your body will actually send less blood to the capillaries in the skin. Therefore, the skin temperature will drop. This will lead to a smaller difference in temperature between the skin and the environment, and therefore less conduction of heat or radiation of heat from the body to the environment. The skin is also, the circulatory system is also involved in protecting the body. So, for example, within your body, you have platelets, which are involved in clotting. You also have white blood cells that are part of your immune response. As you're probably already aware, your heart is four-chambered, but the best way to think about the heart is that it's actually two pumps, not one pump. And let me go through the logic of that. So if you look at your heart, it has a left and a right side, and each side constitutes a pump. So for example, if you look at the right side of your heart in the simplified diagram, you can see the blood is coming back through a vein into the heart. Now it's not shown here, but there's the right atrium. And then from there, the blood will flow into the right ventricle. Okay, so from there, it will flow out through an artery, the pulmonary artery, to the lungs. The blood will pass through the pulmonary capillaries, picking up oxygen. You see the blood is shown as being changing color from blue to red. So we're going from deoxygenated blood to oxygenated blood. The blood then returns to the heart and is returning to the left atrium. Now this circuit here is referred to as the pulmonary circuit, or alternatively as pulmonary circulation. So this is starting on the right side and coming and ending on the left side of the heart. So it's important, we'll come back to this idea, but this is one circuit or pulmonary circulation. And so in order for the blood to move through this, you need a pressure difference. And what's the pressure source? Well, it's the heart. And here we're specifically talking about the right side of the heart. And it's primarily the right ventricle that's contracting, increasing pressure within the pulmonary artery. And then that's essentially the potential energy that's used. That pressure is able to be converted, can be thought of as a kind of potential energy, which is converted to kinetic energy, energy of movement of the fluid. It flows through this pulmonary circuit, and then back to the heart. So that's one circuit, okay? And that's one pump. Now, there's a second pump, which is the left side of the heart, and there's a second circuit, which is the systemic circuit, or also referred to as systemic circulation. So this starts with the blood being collected again in the left atrium. And then this is very schematic here, right? It's not meant to be anatomically correct here in this diagram, but it shows in principle what's going on very well, which is then the blood then exits the right ventricle, which is the chamber below the right atrium. And we'll come back and I'll show a more detailed picture in a second. The blood exits into the aorta, goes to various major arteries and so forth. And of course, this is highly simplified, but just showing you a single capillary bed. There's of course many capillary beds in the systemic circulation. But the blood will go through that, and it's oxygenated because it's returned from the lungs. And now the tissues of your body need oxygen, and so oxygen will diffuse, and the blood will become deoxygenated, shown in blue here, as it leaves these capillaries, and then it returns in veins to the right atrium, so the right side of the heart. So this is another circuit, and this is referred to as the systemic circuit or systemic circulation. And there's another pump for that. What's the force that drives the blood through this? Well, it's the left ventricle, which is very muscular. It contracts, creating an increase in pressure. Again, the pressure can be thought of as a kind of potential energy. That potential energy is changed to kinetic energy or energy of movement. The blood then moves through the systemic circulation and ends up back in the right atrium. So essentially, you have two pumps, right? The left side is one pump that's responsible for the systemic circulation, and then the second pump is on the right side, and that's responsible for creating the pressure that pushes blood through the pulmonary circuit. This is another simplified diagram. It shows the blood vessels. So we have, starting from the heart, we have the aorta, then we have major arteries. So those are relatively large diameter blood vessels. Arterioles are very small diameter blood vessels, and they have smooth muscle associated with them, so they can vasodilate, meaning they can increase in diameter, and they can vasoconstrict, meaning they decrease in diameter. And this is a way for your body to regulate blood flow to various capillary beds. The smallest vessels are the capillaries. So these are very small in diameter. They're about the diameter of a single red blood cell. And so you have these capillary beds in various tissues, such as your skeletal muscle. And it's at the level of the capillaries that you actually have exchange of materials, such as nutrients, such as glucose, and you also have movement of gases, such as oxygen and CO2 by diffusion across the walls of the capillaries and into the interstitial fluid. Remember, the interstitial fluid is the fluid that surrounds the cells of your body, of the tissues of your body. Now, if we continue along, the smallest veins are called venules, and then those bring fluid back into the veins, which are the larger and larger until you arrive at the superior and inferior vena cava, which then bring the blood back to the heart, to the right side of the heart. Now, another point I should make here is that your capillaries in much of your body are leaky, meaning there's a little bit of paracellular transport, meaning or movement of molecules, meaning the molecules can move between cells rather than through cells, which is transcellular transport. And there's a net, we'll talk about this in more detail later, but there's a net movement of water out of the capillaries. And it's quite a bit. So over the course of a day, quite a bit of water leaves your capillaries, and this has to be brought back into the vascular system. And the way your body does that is there's another whole other system of small vessels called the lymphatic system. This is just a reminder of what we've talked about before. You have the blood plasma. Again, as I just said, gases can diffuse quite easily across the walls of the capillaries into the interstitial fluid and then into cells. Other molecules such as glucose require transporters. We've talked about that. And then also there are many capillaries in your body that are leaky, allowing for paracellular transport. And you can have the movement of water together with solutes, such as various salts, electrolytes, such as sodium, potassium, chloride, and so forth. And then the fluid that actually bathes the cells is called interstitial fluid. There is a major difference between interstitial fluid and plasma, which is plasma contains far more protein than the interstitial fluid. In other respects, they're quite similar. The concentrations of the various electrolytes are relatively similar between interstitial fluid and blood plasma. However, proteins are essentially retained within the vascular system. And the significance of that is something quite important. We'll come back to that a little bit later. Now, as I mentioned, there's essentially a net efflux of water from your blood vessels. And if that were allowed to just continue without any collection of that water, then you would have a condition called edema, which is swelling of the tissues of your body. And in fact, you do experience this on a long plane flight. You may experience slight swelling of your feet and your lower legs if you're not able to get up and walk around enough. Normally, you don't have this problem because there's the lymphatic system. And one of the functions of the lymphatic system is to collect this excess fluid. The lymphatic system contains lots of small lymph vessels. These vessels often have smooth muscle associated with them, which can pump the fluid. And these vessels also have one-way valves. And so one of the objects of the lymphatic system is to bring this excess fluid back, and it will actually bring it back to about the level of the neck, just below the neck. There are places where there are ducts in which the lymphatic fluid will then join back with blood within the cardiovascular system. Now, the second function of the lymphatic system is to filter the blood. So there, for example, you're probably quite familiar with the idea that you have lymph nodes. So this lymphatic fluid will flow through lymph nodes, which act as a filter. There are many cells are involved in the immune system, white blood cells and so forth, that can attack bacteria, destroy cellular debris, and so forth. We'll next talk a little bit about the composition of blood. The average adult has a blood volume of about 5 liters. Okay, so the terms artery and vein are based on the direction of blood flow with respect to the heart. So blood leaving the heart is arterial blood, and the blood vessels that leave the heart, that have fluid leaving the heart, are always called arteries. Blood entering the heart is through veins, and this is referred to as venous blood. So again, blood vessels that bring blood back to the heart are always referred to as veins. So note that this definition does not take into account the oxygenation status of the blood. Okay, furthermore, blood has 45% formed elements. So formed elements are things like red blood cells, white blood cells, which we'll talk about in a second, as well as platelets. And then 55% plasma. So plasma is the sort of light yellow colored fluid that contains many solutes and enzymes and so forth. The classic way to measure this in a body from a blood sample is to take blood, add some anticoagulant so that the clotting factors don't start to clot and precipitate out. And then you would centrifuge the blood. And what you'll find when you centrifuge the blood is that these formed elements are heavier than the blood plasma. So they'll end up at the bottom of the centrifuge tube. So for example, here you can see the red blood cells are packed at the bottom of the centrifuge tube. On top of this is what's called a buffy coat, and that's going to be a clear or white layer. within this buffy coat, as you can see in this diagram, you have white blood cells. You may have what else do you have? Platelets, and so those are the major constituents of this buffy coat. A term you'll hear often in medicine is hematocrit. Hematocrit is the percentage of the total blood volume that is packed to red blood cells. So again, you would take a centrifuge, you'd have added anticoagulant already to prevent from the clotting factors from precipitating out. You now have a column, and what you would do is measure the packed red blood cell length, and then divide that by the total length of the full column. So you take the height of the red blood cell column, divided by the height of all components of the blood, then multiply by 100. And then you would arrive at a number which would be the hematocrit. So the normal adult male hematocrit is about 41% to 53%. And for females, hematocrit is 36% to 46%. So notice that the hematocrit is quite a bit higher in adult males compared to females. As I mentioned previously, the plasma, which is that yellowish fluid, contains water and dissolved solutes. Let's talk a little bit about the various solutes in the blood plasma. So there are many proteins, as I mentioned earlier, and those proteins are albumins, globulins, and fibrinogen. Albumin is a type of protein that creates osmotic pressure to help draw water from tissues into capillaries to maintain blood volume and pressure. As I mentioned earlier, the capillaries are very leaky in some parts of the body. And because of their leakiness, there's a net efflux of water. And that's because of blood pressure generated by the heart pushing on the fluid. And so there's a movement of water out through this hydrostatic pressure. Now, to counteract that, you actually have blood proteins, and the number, they all proteins will contribute to the osmotic pressure of the water. Recall that I mentioned earlier that the interstitial fluid is very low in concentration of proteins compared to the blood plasma. So these proteins actually take up quite a bit of the volume and space within the blood plasma, and this means there's less room for water. So the water concentration of the interstitial fluid is higher than the water concentration of the blood plasma. So this means that water will now move down its concentration gradient from the interstitial fluid to the blood plasma. And it's moving across membranes, so that's referred to as osmotic movement or osmosis of water. So water moves by osmosis from the interstitial fluid into the blood plasma. And this is quite a large effect, but it doesn't completely counteract the effect of the blood pressure pushing water out in the opposite direction. So overall, there's a net efflux of water from the blood plasma and into the interstitial fluid. In addition to albumin, there are two other major plasma protein types. There's globulins. There are alpha and beta globulins. These transport lipids and fat-soluble vitamins. So recall that you have your lipids are necessary for plasma membrane. There are certain fat-soluble vitamins that are necessary for your cells. And also you have hormones, certain hormones such as steroid hormones, such as the sex hormones, stress hormones such as cortisol. Now these are all lipid-soluble hormones because they're derived from cholesterol. Now, in order to transport these substances, well, it's very difficult because they're not, they are soluble in water, but not to a very high degree. So in order to effectively transport these sorts of molecules, they need transport mechanism, which include these special globulins, which are proteins that are specifically designed to transport lipids. In other words, they're going to be proteins that are hydrophilic, but they'll have certain regions that are hydrophobic. And so those hydrophobic regions will associate well with lipids such as cholesterol, and then they'll be, and steroid hormones, and they'll be able to transport these lipids around your body. In addition to alpha and beta globulins, which are essentially transport proteins for lipids and fat-soluble vitamins, you have gamma globulins. These are antibodies that function in immunity. Another major class of plasma protein is fibrinogen. Fibrinogen helps in clotting, and what happens is it becomes insoluble threads of fibrin, and that helps to form the actual clot. Another term I should bring up here is the term serum. So far we've talking about blood plasma. So blood plasma is water plus solutes. And the way you would study that and measure that is by, as I mentioned, you would add an anticoagulant to prevent the blood from clotting. Now, if you allow the blood to clot, then what will happen is the fibrinogen will essentially precipitate out as fibrin. And then you'll have a clear fluid that's very similar to blood plasma. It simply now is lacking these clotting agents, the fibrinogen. And that's referred to as serum. In this class, we've mentioned the idea of homeostasis. We used the example of the homeostatic mechanisms that help maintain blood glucose levels within a certain range of values. Another regulated variable is plasma volume. It turns out plasma volume is extremely important for your health. Recall that I've just talked previously about the idea of blood pressure as being the force that pushes blood through the cardiovascular system, through the arteries, through the capillaries, and through the veins. Without sufficient volume, you won't have sufficient blood pressure. There needs to be something for the heart to act on. And so plasma volume is extremely important, and it's a regulated variable. So you have a number of regulatory mechanisms that maintain plasma volume, and this helps to maintain blood pressure. So if you have a decrease in plasma volume, say due to dehydration, then you might expect to have a decrease in blood pressure. But your body counteracts that very quickly and maintains blood pressure. One mechanism, and there are multiple ones, but the one I'll mention here is the fact that you have osmoreceptors in the hypothalamus, which is part of the brain. Within there, these neurons that are osmoreceptors will release antidiuretic hormone, or ADH. And the way this works is the neuron cell bodies are actually in the hypothalamus, but they send axons to the posterior pituitary, which is where they'll actually release the antidiuretic hormone into the blood. So if the osmolality of your blood becomes too high, meaning that you're perhaps dehydrated and the blood is becoming more concentrated, then this will stimulate an increase in antidiuretic hormone release from the posterior pituitary. And the ADH, or antidiuretic hormone, then acts at the level of the kidneys to promote fluid reabsorption by the kidneys. So in the kidneys, you have fluid from the blood entering into these kidney tubules as a fluid called filtrate, and that filtrate contains a lot of water. But then your kidney tubules have these mechanisms for taking up or reabsorbing water back into the body, so that ends it up back into the interstitial fluid and then into capillaries again. So ADH promotes this reabsorption of water, so you don't lose too much. So if you have an example of dehydration, these osmoreceptors in the hypothalamus would be activated. They'd release more ADH from the posterior pituitary. This would act on the kidney to increase fluid reabsorption, and therefore your urine would be more concentrated. Now, one thing I should point out is your body's continually releasing ADH. It's not that it just turns on and turns off. It's just that in a situation such as dehydration, the amount of ADH might increase by 4 or 5-fold. It would increase by a large factor, and that would help to reabsorb water. As we talked about towards the very beginning of this class, the whole concept of homeostasis originated from measurements of blood plasma in particular, and so it's not surprising, because blood is essentially essential for transporting nutrients to your cells, bringing waste away from your cells, and so forth. So it's not surprising. But a number of these things that we'll mention next are actually under fairly tight control. So as I mentioned, blood volume, that's under tight control, homeostatic control. Blood osmolality is roughly about 300 milliosmoles per liter. Blood pH is around 7.4. You also, if you look in your blood, you'll find a certain number of enzymes. For example, there's creatine phosphokinase. This is an enzyme that's found in muscle, and it's involved in the reaction that allows phosphocreatine to donate a phosphate group to adenosine diphosphate, creating ATP. There's a bit of lactate dehydrogenase. This is an enzyme that's very important in the conversion between pyruvate and lactic acid. And back, it's a reversible enzyme. It can go work in both directions. Phosphatases are enzymes that will remove phosphate groups or inorganic phosphate from proteins. As I mentioned earlier, hematocrit's very important. So it's another thing you'd measure in a lab clinical setting. And it's a little bit higher in males than females. Hemoglobin is a type of protein that can bind with oxygen. And so again, that's something you might measure in a blood sample. Another thing that would be important for health would be the red blood cell count. And then, of course, white blood cell count also. White blood cells are involved in the immune response. And again, these are just a few things you might see in a blood test. You might measure testosterone. Note that males have far more testosterone than females. You'd have certain other hormones, such as adrenocorticotropic hormone, or ACTH. Now, this is a hormone that's released by the anterior pituitary gland, and it essentially leads to an increase in the release of cortisol from the adrenal glands. Growth hormones, important for growth in children, growth of the long bones, so that increase in height and increases in muscle mass. And adults, of course, also need growth hormone for restoration of muscle after exercise, restoration of the body during sleep. Insulin, we've talked about, it's a hormone that's involved in regulation of glucose concentration in the blood. And we've talked about some of these ions in the past. So bicarbonate is an important ion. It's a negative anion. It can combine with hydrogen ion to form carbonic acid. So it's part of what's called a bicarbonate buffer in your blood. Calcium, we've mentioned. Calcium's importance for things like release of neurotransmitter at synaptic bouton or axon terminals. Chloride, potassium, and sodium are all involved in the resting membrane potential of neurons as well as muscle cells. And then we have other organic molecules. Cholesterol. Cholesterol is an important component of your cell membranes. It helps to maintain fluidity of the cell membrane. You have glucose, which is a major energy source. You find that in your blood. Lactic acid is also found in the blood. Protein, various proteins. Triglycerides. So remember, triglyceride is glycerol plus 3 fatty acids. You have certain waste products found in your blood, such as urea. It's a nitrogen-containing waste product, and uric acid is another nitrogen-containing waste product. We've talked a little bit about the formed elements in the blood that consists of cellular elements as well as platelets. One of the formed elements are red blood cells, also called erythrocytes. These are shaped as flattened biconcave discs. They carry oxygen. Red blood cells are a little bit unusual in that they lack nuclei and they don't have mitochondria. Because of the lack of mitochondria, they can only create ATP for their cellular processes through anaerobic metabolism. They have a limited lifespan of about 120 days. Note that that's much shorter than most cells, and that's probably because of the lack of the nuclei. And remember, the nucleus is responsible for things like directing protein synthesis through gene expression. Each red blood cells contain millions of hemoglobin molecules. Associated with each hemoglobin is the iron heme, which is recycled in the liver and the spleen. It's carried by the protein transferrin into the blood to the red bone marrow. So red bone marrow is where the red blood cells are actually generated. And there are some conditions that can happen. So for example, anemia is going to be either an abnormally low hemoglobin count or an abnormally low red blood count. And the most common type of anemia is due to an iron deficiency. And how would you have that iron deficiency? Well, one way that you can have this is through blood loss. Another way you can have an iron deficiency is through an inability to absorb iron. The biconcave shape of a red blood cell is thought to have two functions. One is to increase the surface area for diffusion of oxygen and CO2, so that's useful. And the other thing is these blood cells have to squeeze through very small capillaries. So if they're perfectly round, they might get stuck. Having this biconcave shape allows them to fold a little bit and sort of squeeze through very small capillaries and therefore not get stuck. In addition to the red blood cells, other formed elements are white blood cells or leukocytes. These are nuclei and they also have mitochondria, so they're a bit more long-lived than red blood cells potentially. And these are able to move around the body in an amoeboid fashion. So what they do is they circulate in the blood. They may take up residence in various organs, but many of them are in the blood. And when they receive a signal that there's been an attack on the body, say you get a splinter, then there would be cytokines, which are signaling molecules that are released in the immediate area, such as histamine. These would signal to some of these leukocytes that they should enter into the tissue that's been infected. And so they'll pass from the blood plasma through the capillary walls, which we'll recall are a little bit leaky, so they can pass through the spaces between the endothelial cells that make up the capillary walls. They'll enter into the tissue, they'll find cellular debris, they'll find invading bacteria and things like that, and they'll start to phagocytize them. So they engulf them, or at least some of these leukocytes can do phagocytosis. They can engulf these bacteria and then essentially digest them. There are different types of leukocytes. There are granular leukocytes, which comes to the fact that when you stain them, you see these grains within their bodies, and these include neutrophils, eosinophils, and basophils. And then there are agranular leukocytes. So A is a negation, so these don't have clearly visible granules. And these are monocytes and lymphocytes. This is an illustration from your book showing the various types. I've circled granular leukocytes. These would be the neutrophils, the eosinophils, and the basophils. The agranular leukocytes are the lymphocytes and monocytes. Now, in addition to them, you also have platelets and erythrocytes. Recall that erythrocytes are red blood cells. So for the purposes of this course, we're just going to focus on a few of these elements. So the neutrophils, these are phagocytotic. So again, they would enter into an area of infection and they would find invading bacteria and engulf them through this process of phagocytosis. They're also able to do other things, such as release cytokines in a local area of infection. And therefore, this would cause vasodilation, bringing more blood flow to an area of infection. So for example, again, if you had a splinter in your skin, you might see that that area will swell up a little bit, and that's because of edema due to the increased blood flow to that region. And they'll also attract other white blood cells, essentially, to the battle. So that's another type of cytokine they would release. Monocytes is another type we'll focus on here. So this is another sort of phagocytotic cell. And it does essentially some of the similar things that neutrophils do. They can also release cytokines in a local area, increasing blood flow but by causing vasodilation. And they'll also release other cytokines that will essentially recruit more white blood cells to the area, essentially to the battle against the invading bacteria. And again, they would also, as the neutrophils do, they also would break down cellular debris from dead cells. Finally, we'll also focus on a little bit on lymphocytes. You should simply know that these are, among the lymphocytes, there's different types, but certain lymphocytes are involved in the production of antibodies. which are proteins that help in the fight against an infection. Moving on to platelets. So platelets do not have nuclei. So they're essentially acellular they have cell fragments, I should say. And they don't survive very long. They only survive about 5 to 9 days. But they're very important because they're involved in clotting. And we've already mentioned erythrocytes or red blood cells. The third category of formed element are the platelets, or these are also called thrombocytes. They're the smallest form element. They're just fragments of a larger cell called a megakaryocyte. These lack nuclei, and for this reason, they're very short-lived. They only live about 5 to 9 days. They're involved in clotting together with other chemicals and fibrinogen, which we mentioned earlier. That was another plasma. That was a plasma protein. They can release serotonin, which stimulates vasoconstriction. So in an area where you've had a break in the vascular system, you'd want to have less blood flow so that you lose less blood. And so releasing serotonin, which would act in the local area to cause vasoconstriction, would be important. This is a summary table from your book. Note that I've put some of these components in bold. These are the ones you should know. An erythrocyte is a red blood cell. You should know leukocytes, or white blood cells. Granulocytes are these formed elements that have white blood cells that are white blood cells that can have clearly visible granules in them. Among the granulocytes, we'll just focus on neutrophils. You don't have to know eosinophils or basophils. So the neutrophils, as I mentioned, are involved in the immune response. They're phagocytotic, and they can also release cytokines, which have a profound effect locally in terms of vasodilation and also in terms of recruiting more white blood cells to the area. There's also agranulocytes. These are essentially white blood cells that, or leukocytes that do not have clearly visible granules in them. And we'll focus on the monocytes. These are phagocytotic. They have very similar functions to the neutrophils. They can also release cytokines that induce vasodilation, and they can also release cytokines that recruit more white blood cells to an area. But lymphocytes are important also in immune function, and some lymphocytes will generate antibodies. Platelets, also called thrombocytes, are involved in clotting. They release serotonin, which causes vasoconstriction. So both of those factors are helpful when you have a breach or break in the vascular system. We're going to skip this section on hematopoiesis. We're also going to skip the section on red blood cell antigens and blood type. And finally, we're also skipping the section on blood clotting. Okay, in this next section, we're going to go in a lot of detail into understanding both the structure of the heart and also the function of the heart. As we explore the structure of the heart, keep in mind that we have the two types of circulation, the pulmonary circulation and systemic circulation. Blood returns to the heart in the right atrium. So blood comes back from the systemic circuit, from the upper half of the body, the head and so forth, through the superior vena cava. Note that the blood is blue. It's arriving in the right atrium. Blood is also returning from the various capillary beds of the lower half of the body through the inferior vena cava into the right atrium. Again, it's blue blood. It then passes through what's called an atrioventricular valve between the atrium and the ventricle. In this case, it's on the right side of the heart, so that's the right atrioventricular valve. The blood is now in the right ventricle. From there, it passes through the pulmonary valve, which is also could be referred to as a semilunar valve or the right semilunar valve. It goes into the pulmonary trunk, and from there into the pulmonary arteries that go to both lungs on the left and right sides of the body. From there, note the deoxygenated blood is indicated in blue. It picks up oxygen, so it becomes purple, and eventually red, meaning it's oxygenated. And then the blood comes back through the pulmonary veins to the left atrium. Okay, so now we're passing, now the blood is oxygenated, it's red, passes through the left atrioventricular valve or left AV valve into the left ventricle. Note that the left ventricle is very muscular. It then pumps blood through the aorta, which is a semilunar valve, so it's the left semilunar valve. The aorta is the major artery that leaves the heart, and then the aorta does a couple things. One, there are major arteries that move up towards the head, and so this is part of the systemic circulation going up to the head and the upper part of the body. Now the aorta also goes down, it descends, and then it has various major arteries and so forth that come off it. And then these go to smaller capillary beds and so forth. And so the blood then comes back to the heart, again at the right atrium. Now, I mentioned before that the heart is essentially two pumps. The left and the right side are two different separate pumps, but they actually work in a coordinated fashion. So in other words, the atria will contract simultaneously, and after that, the ventricles will contract simultaneously. And how does the body do that? Well, there's actually this thing called a fibrous skeleton that separates the atria from the ventricles. Therefore, they're not one. It's not one continuous muscle from atria to ventricles. There's actually this division, and attached to this fibrous skeleton, you have the atria, which would be above, which aren't shown here, and then below you have the muscles of the ventricles. And it turns out this is very important because in order to have a functional separation where the atria will contract first and then the ventricles, they can't be electrically connected. So recall when we talked about cardiomyocytes or heart cells, that they have electrical connections, which are called gap junctions at the sites of intercalated discs. So intercalated discs are regions where one heart cell contacts another one, and there's strong mechanical connection due to certain proteins there, but there's also gap junctions, which are essentially passages which allow for ions such as sodium, potassium, chloride, and so forth to pass through from one cell to another. Now, if you didn't have this fibrous skeleton, then the whole heart might contract at almost the same time, which is not what should happen functionally. The atria should always contract first, and then the ventricles. And this fibrous skeleton prevents the uncontrolled contraction from going from the atria to the ventricles. And as we'll see, there's a very specific route or pathway for the excitation or depolarization of the atria to move from the atria to the ventricles. This slide simply reiterates what we talked about before, which is that you have two types of circulation: the pulmonary circulation, which is between the heart and the lungs. So the blood is pumped to the lungs via the pulmonary arteries. The blood returns to the heart via the pulmonary veins. And the second type of circulation is the systemic circulation, which is between the heart and the body tissues. The blood is pumped to body tissues via the aorta. The blood returns to the heart via the superior and inferior vena cava. I should note that, and this is a good point to talk about differences between these two types of circulation. Now, if you think about it, the pulmonary circuit's a bit shorter and it's a bit simpler. It's just going to the lungs, whereas the systemic circulation is much more complex with going to many different capillary beds throughout the body. So if you think about it, if you look functionally at the heart, which side of the heart would you expect to be more muscular? Well, it should be the systemic side, the one that pumps the systemic side, which is the left side of the heart. And so if you look anatomically, the left side of the heart is much more muscular than the right side. And so the degree of muscularity of the heart side correlates with the amount of pressure it can generate. So the right side of the heart, which goes to the pulmonary circulation, generates less pressure. And it doesn't really need to generate that much pressure because the resistance through the pulmonary circulation is relatively low. It's a relatively short circuit and so forth. So it doesn't really need to generate that much. Again, you could think of pressure as being potential energy. It doesn't need to generate that much potential energy to have a good flow or movement of blood through the pulmonary circulation. Now, the job of the left side of the heart is much more difficult. It has to be much more muscular. It has to generate much more pressure. In order, again, you can think of pressure as a kind of potential energy. It pushes blood through the circuit that has a lot more resistance. And what's the resistance due to? Well, it's because of the friction between the walls between the moving fluid and the walls of the vascular system. So the resistance is particularly going to be high as you go through the capillaries, which are very thin. And so there's a lot of interaction between the moving fluid and the walls of the capillaries. And so the left heart has to be much more muscular than the right side. This table from your book simply summarizes many things we talked about. So you could look at the differences between the pulmonary and the systemic circulation. The next topic is the valves. So we're going to first tackle the atrioventricular valves, or AV valves. So as I mentioned earlier, these are located between the atria and the ventricles. So you have a right AV valve and a left AV valve. Now, just to make things more complicated in anatomy, we have to name these things, give them alternative names. So between the right atrium, the right ventricle, which is the right AV valve, there's a second name, which is also called the tricuspid valve. And the name is derived from the fact there's actually three cusps, or three of these sort of leaflets to it. So you can see here in this picture, there's three of these. And so if we look here, it's less evident when you look at it from the side, but there's three valves here. There's three leaves to the valve. Now on the other side, you have the left atrioventricular valve, and we can simply call it the left atrioventricular valve. But this is also referred to as the bicuspid valve because it has two cusps. It has two of these leaflets. And in addition to that, just to make things even more complicated, unfortunately, it's also called the mitral valve. And you think, well, what's this crazy name mitral come from? So a mitre is a kind of ceremonial hat worn by bishops. And if you take one of those and flip it upside down, then you'll see that it has these two flaps. And so that looks very much like the mitral valve. It has these two flaps, and so that's the origin of the name mitral valve. So let's look a little more detail at these atrioventricular valves. So they're open. These respond to pressure differences. So if we look at the right side of the heart, when the pressure is higher in the right atrium compared to the right ventricle, then this right atrioventricular valve is open. Okay, so that's the tricuspid valve. And now when the pressure is higher in the right ventricle compared to the right atrium, then these valves shut. So you can think of the fluid sort of pushing on these the cusps of these valves and then pushing them shut. Now there's the possibility that the valve may sort of blow out and evert into the right atrium. Now, to prevent that, you have these things called chordae tendineae, which is the plural. So what these do is these are fibrous cords that attach to these cusps of the valve, and they prevent the valve from everting into the right atrium when there's a higher pressure in the right atrium compared to the right atrium. In addition to these chordae tendineae, there are actually papillary muscles also that contract. So at the time when these valves should be closing, these will contract, and again, that'll also help to prevent the valve from going, opening up by sort of everting into the right atrium. So this is a one-way valve. It's open when the right atrial pressure is higher than the right ventricular pressure. Now, if we look on the other side, it's a very similar situation with a left atrioventricular valve. This valve is open when the left atrial pressure is higher than the right ventricular pressure. Conversely, when the right ventricular pressure is higher than the right atrial pressure, the fluid pushes against these valves and essentially pushing them shut against the leaflets or the cusps, that pushing them shut. And again, you'll have the chordae tendineae, which are these fibrous cords that help prevent the valves from everting. And again, you'll have papillary muscles that will contract at just the right moment to prevent this also. Okay, there are also semilunar valves. These are located between the ventricles and the arteries. that leave the heart. So this term semilunar comes from the fact that the leaflets or cusps of these cells, if you look at them at the right perspective, they look like little half moons. And so there's the pulmonary valve. This is between the right ventricle. Let me see if we can find it here. And this is, so here it is right here. Within the pulmonary trunk, you see the pulmonary valve. And then there's the aortic valve, which actually, unfortunately, I don't see, you can't see it here, but there's the aortic valve, which is just beyond the heart in the very base of the aorta. You have the aortic valve. And so it's between the left ventricle and the aorta. And both of these valves also work based on pressure. So let's think about the pulmonary valve, which is the one we can see here. When the pressure is higher in the pulmonary trunk compared to the right ventricle, then these valves, you can see how they're sort of cup-shaped. So you could imagine the fluids pushing against those cups and sort of forcing the valve shut. And that's exactly how they work. The valves are pushed against, and then they're slammed shut. And the same thing for the aorta. It looks very much similar to the pulmonary valve. So when the aortic pressure is higher than the left ventricular pressure, then the back flow of blood against the valve sort of fills these cups, and the valve is forced shut. Okay, one thing that students are often quite interested in is the heart sounds, right? So these are referred to as lub-dub, or sometimes lub-dup. So these sounds correspond to specific points in the cardiac cycle. So the lub corresponds to the closing of the atrioventricular valve, and this occurs at the beginning of ventricular systole. So we'll talk about that in a second. And then dub corresponds to closing of the semilunar valves, and this occurs at during ventricular diastole. Okay, so let's try and make some sense of that. So the terms systole and diastole refer to, systole has to do with contraction, and diastole has to do with relaxation. And so you have, since both the atria and the ventricles are muscular, you have atrial systole and diastole, and you also have ventricular systole and diastole. Okay, so I'll try and make it clear. If I just say systole and diastole by themselves, I'll be referring to ventricular systole and diastole. But I'll try and make it clear as I speak about this. Let's start here in the cardiac cycle, because it's a very convenient place to start. At this point in the cycle, we have essentially we have atrial diastole, which is shown here, and we also have ventricular diastole. Okay, so both the atria and the ventricles are relaxing. So let's look at the right side of this heart. So blood is flowing back through the superior and inferior vena cava into the right atrium. The pressure is higher in the right ventricle compared to the right atrium compared to the right ventricle. Therefore, blood flow is going to flow from air of high pressure to low pressure. So it's filling the right ventricle. And actually, let's look at this left side. A very similar situation is happening on the left side. So blood is flowing back through from the pulmonary circulation through the pulmonary veins into the left atrium. The left atrial pressure is higher than the left ventricular pressure. Therefore, the left atrial ventricular valve, also called the mitral valve, is open. And so blood is flowing in. So essentially everything's relaxing and blood is flowing in. Now, we're still in a period of ventricular diastole. So that means the ventricles are still relaxing. But what happens next is the atria will contract. This is atrial systole that begins. Remember, the atria are muscular, so they will push blood into the right and left ventricles. And this is often referred to as an atrial kick. What they're doing is the ventricles are already filling with blood, but we want to push even more blood into them. And so we sort of kick more blood in. And this contributes a little more blood into the ventricles, and that's useful because then when the ventricles contract, more blood can be pushed out during the ejection phase of the cardiac cycle. Okay, so this is the next thing that occurs. Now, eventually, the atria go into diastole. They start to relax. And then we go into ventricular systole, which is shown as a slightly darker green here. So the ventricles will begin to contract. And during this phase, the pressure will increase in the right and left ventricles. And so the pressure difference will now reverse. So the pressure will be higher in the ventricles compared to the atria. And remember, these valves respond to pressure differences. So the blood will push against those valves, and they'll shut. And remember, we have the chordae tendineae, which are those heartstrings, and the papillary muscles. These prevent the eversion of these atrioventricular valves into the right and left atria. And so that's when you get your first heart sound, the lub. So lub, what is it caused by? Well, the valves hitting each other are actually, that's kind of silent. But what happens, the various flaps or leaflets of the valves hitting each other, that doesn't actually generate the sound. What actually happens is you had blood flowing in one direction, and then suddenly as the pressure difference changes and the valves shut, then the direction of movement of the fluid changes, and there's kind of a reverberation because the fluid is moving, but then it sort of comes in contact with the valves, which stop its movement. So there's a kind of reverberation or vibration, and that's what actually causes the sound. This vibration, which is due to this change in direction and so forth, and flow and turbulent flow of the water, of the fluid within the lungs, of the heart. So that's the first heart sound. So now the ventricles are contracting. We'll come back to this and talk in a little more detail to some of these things later. But so now you have the ventricles contracting, pressure increases. You'll get to the point now where the pressure is higher in the right ventricle compared to the pulmonary trunk. The pressure is higher in the left ventricle compared to the aorta. So pressure, because these valves are also open in response to pressure, these valves will open. So the opening of the valves, you'll notice, is silent. They've just opened and blood flows out. There's no sound. Now, what happens eventually, so this is the ejection phase. So blood is being ejected into the pulmonary circuit from the right ventricle into the pulmonary arteries and so forth. And blood is being ejected into the aorta from the left ventricle. Now, eventually, the ventricles will go into diastole, meaning they'll relax. So we're switching from this dark green to a lighter green. So as they relax, what will happen is the pressure will actually reverse again. So the pressure is now, if we take the left side of the heart, the pressure in the aorta is actually higher than the pressure in the left ventricle. And that may seem sort of crazy, but the reason is because the arteries are very elastic. So they'll expand a bit in diameter. So as they're doing that, it's, again, you can think of a kind of a potential energy. They're pushing the walls of these very elastic walls out. And eventually, as the left ventricle starts to relax, then those, due to the elasticity, they'll have elastic recoil, and the arteries and the aorta and other major arteries will start to constrict a bit as they just recoil backwards to their normal diameter. And that helps maintain the pressure quite high in the aorta. So the aortic pressure is higher than that of the left ventricle. And remember, if we think about the semilunar valves, remember how they're cup-shaped? So the blood would flow into those cups, slamming those valves shut. And so that's what happens on the left side. And exactly analogous thing happens on the right side. The pressure is higher in the pulmonary trunk compared to the right ventricle. Therefore, the blood will push against those cusps or leaflets of the valve, the pulmonary valve, slamming it shut. Now, again, we've had this sort of reversal of direction of fluid and a bit of turbulent flow, and that causes a vibration of the structures within the heart. And then you get the second heart sound, which is the dub sound. So the dub sound would correspond to the beginning of, essentially, it occurs near the beginning of ventricular diastole. Whereas the lub sound occurs towards the beginning, or could mark the beginning of ventricular systole, okay? And eventually we go back to the original point where now eventually the ventricles relax so much that their pressure is now lower than the atrial pressure on both sides of the heart. And because of that, the atrioventricular valves open. That's a silent event, and blood flows into the right and left atria. And again, and then we can continue all around and around in this cardiac cycle. But that's the origin of the two sounds. So lub corresponds to the closing of the atrioventricular valves, and that occurs essentially during ventricular systole. So we could think of that as marking the beginning of ventricular systole. Because, and it, again, occurs because the pressure is now higher in the ventricles compared to the atria, so that slams those valves shut. And the dub occurs is due to the closing of the semilunar valves, and this occurs ventricular diastole, and that's because the residual pressure in the aorta and the pulmonary artery are now higher than the corresponding left and right ventricles. So far, we talked about what happens, what sort of heart sounds you'd have in a normal, healthy individual. But of course, it's also interesting to look at what might happen in some disease states. So there are heart sounds called heart murmurs. These are abnormal heart sounds produced by abnormal blood flow through the heart. And these are often caused by defective heart valves. So one condition, for example, is mitral stenosis. Now, stenosis refers to the narrowing of something. So this occurs when the mitral valve calcifies. This impairs the flow between the left atrium and the ventricle. So let's look at that. So here we have the mitral valve stenosis. So it's been calcified a bit. And so this impairs blood flow from the right atrium, so the left atrium into the left ventricle. Remember, the blood in the left atrium has just come back from the pulmonary circulation. So it's coming back from the lungs. And normally it should flow very easily into the left ventricle. So what happens if the blood doesn't flow as easily into the left ventricle? Well, the blood can it can back up a bit, and this will increase the pressure. If there's more volume here of blood, then there's an increased pressure in the left atrium. And what will that do? Well, it'll have a sort of backwards effect on the pulmonary circuit. It'll increase the amount of pressure needed for the blood to flow through. So the right ventricle will adapt by growing thicker, and it'll increase the pressure as it pushes the blood through the pulmonary circuit. So none of this is good. It leads to pulmonary hypertension. So higher pressure within the pulmonary circulation can lead to things like edema. So remember what I mentioned, the fact that blood pressure itself can push fluid through the capillary walls, through that endothelium. So you have fluid ending up in the tissue, and that's referred to as edema, the swelling. And in extreme cases in the lungs, you can actually get, you can actually get fluid flowing into the alveoli, which is the little air sacs where diffusion of oxygen and CO2 occur. So it can get quite extreme and quite bad from a medical point of view if you have this pulmonary hypertension. Another condition that would lead to a heart murmur are incompetent valves. These are valves that don't close properly. Recall that the mitral valve, which is the left atrial ventricular valve, should close when the left ventricular pressure is higher than the left atrial pressure. But in certain cases, certain individuals, you can have what's called mitral valve prolapse. This occurs when the flaps or the leaflets of the heart's mitral valve prolapse, meaning they invert into the left atrium, as you can see in this picture. So when that occurs, then that valve doesn't shut properly. And so when it comes to the ejection, for example, the high pressure phase, the ejection phase, when the left ventricle is contracting and pushing blood out into the aorta, some blood will actually go back into the left atrium. So again, that could lead to problems with blood flow in terms of the pulmonary circulation. It's also, of course, a little bit less efficient in terms of blood flowing being ejected as the heart contracts. So again, this condition can be due to a problem with the leaflets or flaps of the mitral valve, and potentially could also be due to damage to the papillary muscles on the left side of the heart. Another potential kind of heart defect that would lead to a murmur would be a septal defect. What a septal defect is a hole either between the two ventricles, or it could be a hole between the two atria. So that would be an interventricular septa or interatrial septa. So this allows blood to cross the sides between the two sides of the heart. So of course that's problematic. You'd be mixing blood that's oxygenated, for example, here with blood that's deoxygenated, and then sending that blood through the pulmonary circuit. Now, generally, as I mentioned earlier, the left side of the heart is more muscular than the right side. So generally what will happen is you'll have blood flowing from the left side of the heart into the right side. So this increases the pressure on the right side of the heart. And again, this could increase the pressure through the whole pulmonary circulation. And higher pressure means higher hydrostatic pressure. The blood can push out through the capillaries, not the blood cells, but the fluid. And this could lead to more fluid into interstitial space and pulmonary edema, again, swelling within the lungs. And so we'll end there for this lecture.